All right, welcome back. This is going to be part two. Here we're going to take a look at the basic overall structure of a neuron. Now, with nervous tissue, we have two main types of cells. We have neurons, and we have things called glial cells or neuroglial cells. Neurons are um, uh, they are less abundant than glial cells, but neurons are, you can think, functionally more important. These are what are going to actually transmit what we'll refer to as an action potential. They're the workhorse, if you will, of our nervous system. They're the really important one that we'll focus on a lot. But we'll also talk about our glial cells and what functions they carry out. So our neuron, it's the excitable cell. It has a membrane potential that can be changed quickly, as we'll see and we'll talk about in later parts of this lecture. But for now, we'll focus on the overall structure of our neuron. So, just like any cell, it has a cell body. There's a main portion to our cell. Within that cell body, you have different organelles. You have the nuclei that you generally associate with other types of cells. It has neurofibrils. These are the cytoskeletal components of a neuron that provide it with structure and rigidity, just as the skeleton does in our bodies. It has things called nissl bodies, and these are areas with clusters of rough endoplasmic reticulum. So these areas of rough endoplasmic reticulum that are all aggregated and clumped up together are called nissl bodies. And we know that if it's rough endoplasmic reticulum, you get the rough from the presence of ribosomes. And we know that ribosomes are involved in protein synthesis. And so this is a site of protein synthesis in neurons. We have dendrites and axons. So coming off of different cells, we have things called processes, or these points. And we have two main types on neurons. We have an axon and we have dendrites. Now, dendrites are what are coming into the soma, or the cell body, and bringing in impulses and electrical signals from other cells. So they're bringing them into the neuron. And then the axon is what that signal leaves the cell on. So after it's received, it comes in through the dendrites to the cell body. It is then propagated along the axon toward the next cell or toward whatever target tissue it is going to innervate. Now, at the very beginning of this, let's see. Okay, sorry about that. I got a phone call. All right, I'm back now. So, as I was saying, we have these different processes that come off of our neurons, our axons. At the very beginning portion of our axon, of the neuron, we have a region that is kind of enlarged a little bit. And this is called the axon hillock. And this will become very important whenever we first start talking about our action potential, because it's at this axon hillock that our action potential is originated. The action potential cannot originate at any other place other than the axon hillock. So the axon hillock is the very beginning. We go to the entire slender axon, and then we get to the very end of the axon, and this is known as the axon terminals. And we have a couple of things in there. We have synaptic knobs or synaptic end bulbs, and we have synaptic vesicles. So these synaptic knobs, or these bulbs, are kind of these rounded and large kind of bulbous ends of the synapse. And then within these synaptic knobs or synaptic bulbs, you have synaptic vesicles. And these vesicles are going to contain neurotransmitters that will then be used in our actual action potential between two, at the end of the action potential, transmitting the signal to the next cell. Now, this axon that we've referred to, sometimes in different settings, you can have it be wrapped and covered and coated in this fatty substance known as myelin. And so when you have this long stretch of myelin over the axon, you get something called the axon you get something called the myelin sheath. Now, this myelin sheath has functional implications for our neuron. When there is myelin present, when the myelin sheath is present, your action potential is able to travel more quickly along the axon of the neuron than when you are unmyelinated, so with a neuron that does not have myelin. Okay, and we'll talk specifically about types of cells that myelinate. You have certain ones that myelinate in the central nervous system. You have certain ones that myelinate in the peripheral nervous system. But the myelin is essentially the same thing, and you get the same result either way. Okay, so here's a classical representation of a neuron. You have the large soma, or the large cell body. You have the dendrites coming into the cell body, right? So you have these little projections all out here coming in. You have a large nucleus in the middle, just like any other cell. This is your axon hillock, and you see, as I alluded to a second ago, it's kind of enlarged and kind of wide, a little wider than the rest of your axon. And so it is enlarged, it tapers down to your slender axon here. You can see that this axon is coated in myelin, so it has a myelin sheath. And so this particular neuron here, we would term a myelinated neuron, or a myelinated axon of a neuron, okay? And then at the end, you can see your axon terminals with synaptic knobs, and then within those knobs, you would have synaptic vesicles within them. And again, those synaptic vesicles contain neurotransmitters, and those neurotransmitters will be responsible for communicating the action potential from this neuron to the next neuron, or conveying the message to some target tissue or some target cell. Okay. Later, we will look at different types of neurons in terms of their structure, and these different neurons in terms of structure will have different functions, and they'll be located in different areas throughout the body. Okay, so our axons that we just talked about, which is part of our neuron, sometimes you'll have regions where there's a big grouping of them, and a large grouping and a bundle of axons is referred to as a nerve, okay, or our nerve fibers that we think of. And so the nerves in our body, some may be common to you. You've heard of maybe the median nerve or the femoral nerve or something like this. These are just groupings of axons of individual neurons, okay? So it's just a bunch of axons all grouped together, okay? Later we'll talk about the cell body portion of our neuron. What happens when you get a clumping and an aggregation of cell bodies? You'll call them one thing when you find them in the central nervous system, and you'll call them something different when you find them in the peripheral nervous system. And so it may help you to think of something that is analogous to this nerve. And so if you picture a bunch of straws or pencils or something slender like that, one individual pencil or straw is one axon of a neuron. When you put a bunch of them together and group them, imagine putting a rubber band around them, now they're all together, and so you have lots of individual pencils or individual straws, which are our axons, as a single unit functioning as a nerve. Okay, so that may be somewhat of a way to kind of remember that. Now, here on the right is our nerve, and I put this picture in of a muscle because we have a very similar arrangement in muscles as we do in our nerves. So in our muscles, we have things called epimysium, perimysium, and endomysium, and these are connective tissue structures, connective tissue coverings that either encircle an entire muscle. If they may encircle a fascicle or a grouping of muscle cells or muscle fibers, and then you have endomysium that encompasses a single individual muscle fiber. So when we come to our nerve, we're going to not call it mysium, but we're going to call it neurium. So here we have an epineurium, we have a perineurium, and we have an endoneurium. Okay, so what you can see is this large nerve is surrounded by epineurium, and when you look inside of your fascicles, you see individual nerve fibers, right? So here's an axon that's covered, here's another one, and there's lots of these in there. So with this illustration, you can easily see that one nerve contains lots of different um, nerve fibers, so lots of different axons, right? Different types of nerves, we'll see later, will be some will be purely motor, some will be purely sensory, some will be a combination of the two, um, some will be parasympathetic, some will be sympathetic. But in terms of the direction, nerves can sometimes um, carry information to a target tissue or cell, and then also carry information from a target tissue or cell back to the central nervous system along the same nerve. And the reason it can do both is because you have uh, different axons and different fascicles in here that may be some may be sensory, some may be motor, and so the reason is you get dual types of axons that transmit messages in both ways. So either to the nervous system with afferent or sensory, and then away from the central nervous system with efferent or motor. So that is all neurons. So that's one type of nerve cell, and it's the most common type in the sense that you hear about it the most. Now, the most abundant type of cells are your glial cells. So you actually have more of these neuroglia or glial cells than you do neurons. Okay, these are supporting cells. They serve to nourish neurons. They help in their functioning. They help to regulate function, and we have different ones in the central nervous system, and then we have different ones in the peripheral nervous system. So we'll take a look here at the central nervous system. We have four main ones. We have astrocytes, oligodendrocytes, microglia, and one's called ependymal cells. Astrocytes, the term astrocyte is referring to star-shaped, and so these cells somewhat resemble the shape of a star. And these help to regulate the exchange of ions and different materials between the blood and the brain tissue, and so it helps to maintain something called the blood-brain barrier. These astrocytes, you'll see that they will kind of cover the surface of a cell of the cells lining the blood vessels that prevent things from readily leaving the blood and going into the brain tissue. So with the brain, you can imagine we want to closely regulate and closely control what is going in, and so these astrocytes help to do that. We have oligodendrocytes. This is going to be one of our myelinating cells, specifically in the central nervous system. Now, we'll talk about Schwann cells with the peripheral nervous system. The cells both do the same thing. They both myelinate. Now, the mechanism by which they myelinate is slightly different, and the number of cells that they myelinate is a little bit different as well. Okay, our microglia are phagocytic cells, so they're part of our immune system that will phagocytize and engulf any foreign debris, dead cell parts, things like that, foreign cells that would be within our nervous system. And then we have ones called ependymal cells, and we'll see that these ependymal cells line regions of our nervous system called ventricles, and they make up this structure called the choroid plexus, and these help to filter blood, and by filtering blood, we get something called cerebral spinal fluid, or CSF. And we'll talk later about CSF and how CSF circulates around our brain, circulates around our spinal cord, and serves to protect and kind of cushion these structures. Okay, so those are all central nervous system glial cells. We also have peripheral nervous system glial cells, Schwann cells and satellite cells. Schwann cells are the corresponding cell in the peripheral nervous system where you had oligodendrocytes in the central nervous system. These Schwann cells are going to myelinate axons in your peripheral nervous system, and so peripheral nerves. And they typically myelinate just a single cell per Schwann cell, and they wrap around the axon. So in a little bit, I'll show a picture and an illustration of one. And what you see is this Schwann cell wrapping itself around the axon of the neuron, and that's the myelin itself. And then we have satellite cells that have a similar function to the astrocytes, but only in the peripheral nervous system. So they help to regulate ion and electrolyte balance between different cells. We'll look later that these neurons will have different concentrations of sodium and potassium and other ions inside and outside the cell. And so these satellite cells can help to, at least in part, kind of maintain and regulate that balance. All right, so here you can see different types of neuroglial cells within the central nervous system. So on the left here, astrocytes, you can see they somewhat resemble a star with these projections coming out. Oligodendrocytes, you can see that it is myelinating multiple regions of an axon just from the one cell. You have microglial cells, which are the phagocytic cells of the central nervous system, and then our ependymal cells, which again we said help to maintain, help to produce our cerebrospinal fluid, our CSF, within the ventricles of our brain. And we'll talk about the different types of ventricles that we have. We have 4 of them. And then on the right, if we come over here, we can see a picture of a Schwann cell. Okay, so Schwann cell is the comparable cell to the oligodendrocyte, but only in the peripheral nervous system. And you can see that this cell, if you kind of look at it, you see it just consistently wrapping itself around this axon here. And here you can see the nucleus of that cell. And so all it does is just continually wrap itself around, and by doing so, it produces the myelin. And so you can see this image here. This is dictating the myelin, representing the myelin here. And this is going to help our action potential travel down this neuron, down this axon, very, very quickly. Our ependymal cells, as I said, help to produce our cerebrospinal fluid. And so this is this slide here is depicting the different ventricles of our brain. And so this kind of looks alien-like, kind of like an alien structure. But what it is, it's these hollow spaces within our brain and leading down into our spinal cord and around our spinal cord that contains this fluid called cerebrospinal fluid. So lining these ventricles, you have ependymal cells that make up something called the choroid plexus. And blood will flow through this. It will be filtered. Different ions and whatnot will be taken out, different things will be exchanged, and what you're left with is this relatively clear fluid, this liquid, that circulates through these ventricles. So this is these long ones here, these are called your lateral ventricles. You have 2, you have a left and a right. This kind of circular one here in the middle is called the third ventricle, and you can see it here. And then down here, you have a 4th ventricle, which is way down here. And then from this 4th ventricle, your cerebrospinal fluid will exit, circulate around the brain, and circulate around the spinal cord. This CSF circulates in the subarachnoid space, and we'll talk about the meninges when we get to the when we talk more about the spinal cord. But the meninges are these connective tissue layers and coverings of the spinal cord, and the CSF will circulate beneath one called the arachnoid mater, so in the subarachnoid space. All right, we're going to stop there with part 2, and we'll pick up with part 3 in the next video.